This afternoon we're going to be talking a little bit about phosphorus, and I think there was a couple other talks. There was at least an update about the new Phosphorus Management Tool. I'm going to mention that just a little bit. But I actually kind of got inspired to give this talk. I was asked to speak at Queen Anne's County Agronomy Day this year, and Jenny Rhodes only gave me 25 minutes, short time, and she said, I just want you to tell everyone all they ever need to know about phosphorus. And I was like, talk about an impossible task. You only give me 25 minutes to tell people everything they need to know about phosphorus. And so it went over really well. It was such a blur, I don't even know what I said, but I figured I'd try it again. Here at crop school, I got a little bit longer so I can expand on it a little bit, hopefully talk a little slower. So I'm going to do my best to provide you all you need to know about phosphorus. Yes, we are going to talk about the P-index and the PMT for like a wee little bit, but mostly just talking about phosphorus in general. And then I'm going to show you guys some data from the long-term phosphorus drawdown study that we've been working on. And if you're a regular attendee at this event, you've probably seen some version of this data, maybe not last year, maybe not the year before, but at some point some of this data has been presented. So it may look a little different. Hang on till the end because I think it's going to look a little different than what you've seen it before. But I wanted to share some of that data because we're finally getting it published. Yay! Okay. So let's jump into phosphorus. I don't think I need to tell this audience that phosphorus is really important, right? It's an essential nutrient for plant growth. It's a component of RNA, DNA, ATP, which is the energy currency in the plant, promotes early growth. Sometimes actually the deficiency is different. It's similar to nitrogen because phosphorus is really needed for the assimilation of those nitrates in the plant. And it's also quite mobile within the plant. And so I like to show the purple corn, beautiful. Actually, what I thought, and of course the dose, that not all varieties will actually produce this purple color in a phosphorus deficiency. Only certain varieties actually have the capability to form that purple color. I just thought that was a fun little tidbit that I learned as I was putting this presentation together. So that's phosphorus deficiency. I think we've all heard about this before. All right. Knowing the favorite slide, the phosphorus cycle. I don't know if I've told this story. Some of you who've heard me talk may have heard me tell this story before, but I remember going to bed before my comprehensive exams, like memorizing. I had the phosphorus cycle burned in the back of one eyelid, and then the nitrogen cycle on the back of the other eyelid. So every time I put this slide up, it like brings me back to the day of my comprehensive exams where I tried not to cry as they asked me questions for like 3 hours. So memories come back when I look at this slide. But I like the phosphorus cycle. I think it's a lot simpler than the nitrogen cycle, so that's why I stick with phosphorus. So let's talk about this slide a little bit, right? Everything we need to know about phosphorus. So the blue squares are inputs to the soil, and the green circles are the different components or different forms that phosphorus can be in the soil, right? And then we've got, looking at red, we'll call those rectangles, that are the different loss pathways, okay? So we're going to go through and kind of go through this probably ad nauseum for some of you guys, but I want to do my diligence. So we have a couple different boxes here that are the inputs to the soil. Mineral fertilizers, those inorganic fertilizers, and then we've got our plant residues and animal manures, and those are the organic forms of that phosphorus, right? This is basically the same thing there. Just an example of, fertilizer versus those organic sources. There's some other minor sources, but those are the major ones, the crop residues, inorganic fertilizers, and that organic manures and organic amendments versus talk about as far as inputs. So when we go back to the cycle, we see look at organic sources, the plant residue, and those you're going to see stunted, wilted, discolored, and dead seedlings. Cool, okay? And I teach undergrad, so I break things down a lot simpler for them. It's important to know that this organic pool does not have that arrow going into the crop, right? It has to change forms to that solution or that plant available P form. So this is a really good example of a long-term storage. We love organic matter, right? It's like a sponge. It holds on to that water, slow release over time. Also good for hanging on to that phosphorus. So we've got our organic pool here. We've got our solution phosphorus pool that goes directly up into the plant. And then we have a couple other examples of pools or different forms that phosphorus can be in the soil. Let's see if I put the slide. Right. So that was the slide I thought. We've got our solution pool, just in words, pictures and words. So we have those chemical reactions from those other forms of the phosphorus that slowly replenish that plant available P pool. Okay? So they're held sometimes in those other forms, those other pools of phosphorus, and as the plant draws out that solution P, they get replenished in other forms. And then again, we've got our organic phosphorus. It's not plant available, but it's it's an abundant source of phosphorus in the soil. Okay. So those are those over here. We've got a couple this is over here. We've got our arrows going back and forth again. These forms can be broken down and replenish that solution phosphorus. So we've got our mineral surfaces, right? We've got the surfaces that hold on to the mixture with the phosphorus, and some a little tighter than others, right? So the example here, the rectangle can either be clay, a clay mineral. The charges are right. It wants to hold on and hang on to that phosphorus in the soil. Or when we get into those more acid soils, we got those iron and those aluminum hydroxides that really have a very strong affinity for that phosphorus. And then as we move up into the more alkaline soils, we got that calcium. It also has a similar affinity to hold on to that phosphorus. So this is kind of, we call this sorbed phosphorus. It's being held on, but it can change forms, and it can go back into that solution P and become plant available. All right? So that's this example over here, those kind of mineral surfaces. And these are a little fuzzy because I blew up that picture. But what they say is adsorption and desorption. So that means it's more of a surface relationship. Okay? That phosphorus is kind of on the surface, and it can go back and forth between those pools. But then we move into those kind of secondary compounds. And that's really what we're talking about, like a chunk of phosphorus that's involved, that's seriously involved. They got a strong relationship there. And it's going to take something like dissolution with acid to really break that phosphorus away from this compound that it's formed. It's a very slow process for this phosphorus to become more available into the solution, the soil solution phosphorus. So I wish one day when I'm motivated, I'll remake this figure to kind of really show that better with these arrows. But the important thing to note is that this phosphorus is being held much more tightly, and it's less available, or it's going to take a long time to become more available to this soil solution pool. And this is really important. I'm setting myself up for later when I talk about the drawdown study. What's really important, I should have put this slide back in later, is to show that when we think of a soil test, right, we go out, we stick our probe in the ground, we send it to the lab, and we get a result back. We want to be really clear about what that means. So that result is measuring some portion of this solution P. We're looking at what the make up. But we're not really taking into account these other forms that phosphorus can be held in. So we're going to get back to that later. A little foreshadowing. Burn this picture into the back of your brain like I have. Okay? All right. So when we talk about phosphorus availability, and I'm sure you guys are all familiar with this, the availability of phosphorus is well tied to your pH in the soil, right? We all know pH is our master variable. It controls how available our nutrients are, and I have a beautiful picture in the next slide to show you guys that. But specifically with phosphorus, we start getting out of our ideal pH range, and something is going to tie up that phosphorus and pull it out of that solution P, okay, that solution P pool. Okay, so as we start getting acidic, it's those and a little alkaline, it's that calcium. And so we see our greatest phosphorus availability when we're within our ideal pH range. And there's that beautiful picture. I don't know who I stole this from, but thank you, whoever I stole it from. Google, I think. And so, you know, not only is it phosphorus, which is what I was supposed to talk about today, but, you know, the other nutrients are controlled by that pH range. And so that's really important. That we're sticking in that range, but it's important for phosphorus as well. which I think is really important. You know, phosphorus being one of those micros as well, but I think it's important that we're keeping that in mind with our phosphorus, especially here because, you know, we like to think of, we have very high P in our soils, right? And we know that in the Delmarva. And so, you know, making sure we still have that ideal pH range to keep that phosphorus available. All right, when we talk about issues with the Chesapeake Bay, which I know we're all familiar with, we usually lump nitrogen and phosphorus together, right? That's always what we hear. Nitrogen and phosphorus, yes, nitrogen and phosphorus, that. The point I want to get across is that we lump them together and we talk about them together, but they really behave very differently, okay? And we talk a lot about nitrogen use efficiency, right? Putting out what the crop needs when it needs it. And that's still important for phosphorus, but we have to understand phosphorus, and so you can justify the use of. And so when we're talking about, oh, let's just increase our phosphorus use efficiency, it's not the same principles as it is in nitrogen, okay? And this quote, I know I stole this from Josh McGrath. There is a dubious, if not non-existent relationship between phosphorus use efficiency and phosphorus loss. Phosphorus and nitrogen don't work the same. The only way to decrease loss is through management of timing and placement. This management is going to become important later when I start getting into the Phosphorus Management Tool. So, and that's the focus of, we've all heard about the 4 R's, you know, right rate, right source, right rate, right time, right place. So basically increasing your management, doing a better job. This is kind of the way that we can move forward with our phosphorus management, okay? All right, I'm going to talk a little bit about transport pathways. So we've talked about the source side, right? We've talked about the different forms in the soil, how they can exist, and now I'm going to talk about how they can leave your field, okay? You know, this is what we're all trying to prevent, all right? And I will say, I made these beautiful animations. If anyone steals them from me, I want credit for them, okay? For transmitting virus. So let's go through. We're going to talk about particulate P loss, erosion. We all know about this, right? In this slide, this brown spot is a soil particle. And we've got our phosphorus that's being held on to that aluminum, the iron, the clay surfaces. So our phosphorus is inside that soil particle, right? And we mostly see erosion, sloped landscapes. There's a lot of erosion. And so what happens is we get a rain event, and that whole soil particle moves from its original place to off the field to a body of water, right? This is erosion. And so what do we do? What are management practices that we can combat this specific phosphorus loss pathway, right? Corn maggot damage. Maybe implement no-till, minimize your tillage. Any practices that decrease erosion. I put an asterisk here because when I go to the next slide, this is going to be different, okay? So particulate P losses, that P that's in a soil particle, we want to decrease some kind of minimum or no-till to cut down this specific P loss pathway, all right? Let's go to the next one. Now this is runoff P. Now this P is different. In this slide, this brown blob is a pile of manure. I know, I had to label them because they look quite similar from slide to slide. Just so we're clear, in this slide, this is now a pile of manure. It could be fertilizer. It's some other kind of source, okay? And this phosphorus, it's not being held very tightly in there. So we're still in a sloped landscape, right? Because we're going to see that water move off the field. But this time, the source is not the actual soil. It can be, but in my picture right here, it's some other kind of source, fertilizer or manure. So then what happens? We get a rain event. That phosphorus is not really being held very tight in this manure or fertilizer. So the individual phosphorus, that phosphorus is now soluble. It moves independently off the field from that source into a body of water, okay? So now we're going to talk about what is the management that we can do for this. All of a sudden that looks a little not from last time, right? Now, well, the first obvious would be try to decrease the application of that source, right? Don't put it down. That may not be the case. You may have to. So what can you do? Now your management becomes the opposite. You want to increase your tillage. You want to get that source, you want to get that soluble phosphorus incorporated, interacting with some soil to hold on to it, okay? So we've talked about 2 P loss pathways, and the management options are completely opposite, okay? So we really need to be sure we're identifying what our transport pathway is. And so this example here was a source. Fertilizer, some kind of source that you put on top of the soil. But it's important to know that the soil itself can also be the source, okay? So we get this situation, especially if we're doing, long-term no-till and we're getting a lot of P application on the surface, where we get this saturation, right? I like to think of the soil as a cup, as a glass. And every time you're putting some kind of an application, you're filling up that glass. And eventually, you fill your glass and it starts running over, right? The soil is exactly the same. There is some finite capacity for holding on to that phosphorus, where if there's continued application, it has to go except be lost. And that's where we get in the situation where this phosphorus that's normally held very tightly in the soil becomes very soluble. Bring them on right out. And so when we have these surface soils that have very high concentration, P concentration, and they're saturated, interact with some precipitation on a sloped surface, and all of a sudden you've got soluble P loss. And so, this has been demonstrated in the literature that when we look at that saturation and we compare it to measured losses of P, so we have this repeated relationship. So as we start getting saturated, especially just in that shallow surface, an increase of that dissolved P, that soluble P in the runoff, all right? So those are our surface loss pathways. Particulate, the whole soil particle moves, or this soluble P dissolves out of its products and moves with water. We also have this surface phosphorus loss that we're all probably pretty familiar with if you're here on the Delmarva, right? So this happens in very resistant to those emissions, which we wonderfully happen to have here on the shore, right? You've got a very flat landscape, check. You typically have artificial drainage and a high water table, check. And you've got low clay content in your soil, high, very sandy soil, check. We have all of the conditions perfect here for this P loss pathway, all right? So let's look at what happens. So you've got your surface P application, or fertilizer, whatever. You apply it, and some of it moves down into the soil, and it eventually bumps, clay particle, aluminum, and iron out, right? So over time, some of your phosphorus is being picked up and held in that soil. But you eventually get to a situation, which we're at in some places here on the shore, where that soil is saturated, right? Your glass is full. You've filled your glass with phosphorus, but you keep applying more. So what ends up happening? We've got very sandy soils. You get a precipitation event. It moves very quickly down through the soil profile, but it's taking that soluble P with it. So it moves down through the profile and then is shuttled very quickly to that nearby ditch, tile, whatever, all right? And so this is kind of the third P loss pathway that, they show it on the phosphorus cycle because they have to. But if you talk to someone who lives in a different part of the country, they're not even worried about it. It's taken an issue, but it's a serious issue here where we live, okay? And so that's why I'm making sure that I want to talk about it. And so some of our management practices that we can do to kind of combat that, again, increase that contact between soil that's not saturated, so you got to go a little deeper with your tillage, and that soluble P source, okay? So I hope I've made the point with my little asterisk down here that your management to deal with that phosphorus loss is going to be different depending on what your transport pathway is, okay? All right, so 2009, they were revising one of the reasons that people are these standards for, water quality, and we all had the P index here by now, right? Maryland had the P index. We're all doing nutrient management planning, but you know, EPA decided, well, I don't know, a P-index thing. I'm not really sure if it's worth it. We probably shouldn't research it anymore. So why don't we just stop using the P-index? And let's just use soil test P, right? Everyone's already taken their soil samples for their nutrient management plan. So let's just skip that whole phosphorus index thing, and let's just use the soil test P because it's easy, it's there, everybody's got it, no one needs training for that. That's what we're going to do, all right? So I'm going to show you a couple slides. I will preface that from the first slide, I can kind of see where they thought this, okay? About the food. Defend them for a second. When you look at this graph, you're going to say, I can kind of see what they mean. But I'm going to pick this graph apart, and I'm going to show you that they were wrong, all right? My favorite thing to do is prove everybody wrong, right? My husband loves it. I'm always telling him he's wrong, you know? So, all right. So when you look at this graph, and I'm going to show a different form of this graph over and over again, all right? So on our x-axis here, we've got our Malic 3 soil P. How much phosphorus is in your soil, all right? And then when we look at the y-axis, we've got measured P loss, all right? So this is, they actually put it in the edge of the field, how much phosphorus was lost, all right? And so we plot this out, and right, you can look at this and say, well, that looks pretty darn good. I guess I can see where they're coming from. We've got a pretty strong relationship here. We've got our optimum P over here. I don't know, I think this is pointing at 30, 50, something like that, right? And so you say, all right, that looks pretty good. I guess I can see where EPA was going with this. But my concern is this area right down here. Even though we've got a nice strong relationship going on here, when you look at this area down here and you're looking at P loss, measured P loss, whether you have a soil over here at about 30 or over here at about 200, you're seeing about the same amount of phosphorus loss, right? And I just think that that's an interesting point to note because that's a pretty huge range, that's a pretty large range of soil test P there, okay? I don't know. First glance, this looks nice, but when you start picking it apart, it starts falling apart a little bit, all right? So let's move on. I love this slide. I'm so glad Doug Beagle let me steal this from him. Okay, so let's look at this watershed. This is a very small watershed. The dashed lines show the outline of that watershed up in the middle of Pennsylvania. The folks that work up at ARS up there have studied the bejesus out of this watershed. They know everything about this watershed, all right? So this is our small watershed, and there's the water, and it's draining this way, all right? And they have determined through their years of studying this, that these red highlighted areas on the left side here, this area of the watershed is what is responsible for 90% of the phosphorus that's being lost from that whole watershed. 10% of the area in that watershed is responsible for 90% of the phosphorus that's being lost at the outlet from that whole watershed, all right? Let's look over here at the right side. Same watershed, same dashed line, the blue line going through the middle. Now we're looking at the individual field overlaid by color with the soil test P, right? P concentration in the soil. So when you kind of like look at one, look at the other, look at one, look at the other. You look at where these red spots are and how they correspond with the areas over here. Some of them are the same, right? There's some areas that are part of the 10%, right, that fall in this high category. Some of them are only in the medium category. And then you've got this oven mitt looking field over here that's scoring high, but not contributing anything to the phosphorus that's being lost from that watershed. So now, is it fair, is it right to tell the person who manages this field that they can't apply any phosphorus to that field?
The mean number of aphids per 6 feet, and you notice this axis only goes up to 20, so we really weren't finding that many aphids.
Right to use soil testing? If I was that guy, I'd say no. So maybe soil testing isn't giving us the whole story here. All right, so let's go back to this graph again. Now, I neglected to mention before, because I was trying to make you guys go along with what the EPA said, that this is the same points we just looked at. I'm just going to add some more. So these red dots represent fields where there's been no recent P input. All right. Fields haven't gotten any phosphorus recently, went out and took soil samples and measured phosphorus loss. Fields with no recent P inputs. We start adding a couple more points. I should have changed those colors. We start adding a couple more fields where there's been either inorganic fertilizer or some type of manure applied, and such load just falls apart, right? And we've got this whole cluster of fields, notice the threshold, that have the same soil test P, same P concentration in the soil, but we've got the whole range of phosphorus loss. And I do want to point out, because I know you guys have been looking at numbers all day, and 10 here and 0.2 there, you know, decimal points of phosphorus loss mean huge amounts environmentally. So to you, you might look at that and say, well, I really don't care if I lose a pound of phosphorus per acre from my field. To me, that doesn't matter. But environmental-wise, just that little amount of phosphorus can have a huge impact on water quality. So I don't want you to get the wrong impression that you're looking at this graph and you're saying, 2.4, who really cares about that? I know maybe to you, economically, you might not care, but environmental-wise, that's a hugely important number. All right. Oh, another good slide that I stole from someone else. All right. So that was source, right? We talked about source here. When you throw in some fields where you've got P applied recently, it messes up this beautiful curve, right? So that was source, first thing. All right. Now we're back to that same watershed again. If you can't tell, these fields are kind of planted on the contour, so the slope is coming down here, and here's that stream, right? This is just one of those fields in that same watershed up in PA, all right? And so they put these little monitoring areas to catch what was coming off of each of these fields, all right? All right, so let's look at the characteristics of these fields. So we've got 1, 2, 3, these 3 fields, and so the orange bars represent soil test P. They took a soil sample, and this is what they came back with. So we've got high P up here, you know, still high P down in the second field. The third field, it's still high, but we're going to call it low relative to the other ones, all right? Lowest. It had the lowest P concentration of these 3 fields. So again, if we were just going by soil test, and you were in the situation, you're a producer, and you had to get rid of that manure, if you were just going by soil test, you'd say, well, this one has the lowest P, that's where I'm going to put my manure, all right? Let's see how that turns out. So in this example, though, they said, you know what, we're going to put the phosphorus in the upper upland fields. We're not going to put anything down here. Even though, if you're using soil test P, this is where you would put that manure, all right? Now we're looking at runoff in liters. That's the actual quantity of water that came off of those fields, all right? Astronomical amount that came off of this lower field, right? Not very much coming off of those upper fields. Then when we finally add in how much phosphorus was in that, we're looking at 8 times the amount in this lower field that you would have put all of your manure down, again, if you had used soil test P as your decision-making tool. All right? And again, I mentioned decimal points can have a huge impact environmentally. So we're going less than 1 to 1, and now we've jumped up to 8. Huge losses. And that was with not applying any phosphorus. Right? And so this is telling us transport is important as well. So we've talked about source, and we've talked about transport.
They had 3 insecticide seed treatments, and they had no treatment control, which is this purple line. And so the insecticide seed treatments are the blue.
Source areas. These are areas in a watershed that have both a source and a transport pathway of phosphorus. You need both, okay? You need both to have a phosphorus loss. Remember our oven mitt over here? The oven mitt field that was red because it had high soil SP? It's got high source, but but it doesn't have any transport. It's not connected to that body of water. It's not responsible for any of that phosphorus being lost to that body of water. All right? So it's really important that you have both, a source and a transport pathway. You can only have one. Not a critical source area. You're not the 10%. Okay? And so
And you got less.
really the purpose. That's why we have the phosphorus index. The phosphorus index, when it works, when it's updated and when it works, it's identifying
Many years, over a couple of years, so this is the 2013 data.
I think that this is really important. I think that this is important from a water quality perspective as well.
So if it's red,
If there is some lump sum of cost share money out there to implement better,
some negative difference.
no offense, but why are we giving that money to the oven mitt field? They're not responsible for the loss. Why don't we focus that money on these critical source areas, get more bang for our buck, right? That just makes sense to me. I'm no businessman, but that makes sense to me. All right, so now we come back to our
Improvements really.
and on x-axis, we changed that from soil test P, which we decided not a good decision-making tool, and we use our P index values. And when we plot those against phosphorus loss, measured phosphorus loss, we see that nice, beautiful curve return. We've even got our points in there that have had different manure applied to them, and they all line up nice. So that phosphorus index really does better describe that potential for phosphorus loss, okay? And so the P index has a couple different versions. It's gone through a couple iterations since its inception in 1993. So the first P index, they said, all right, we need source and we need transport. Why don't we just add them all up, right? And so that's what we've got here. All of the variables that represent your source summed up and added to all of your variables that represent your transport. And that was your P index score. Okay, I gotta go faster. All right. And then we had a generation where they said, you know what, I don't think that math is working out right. So I'm gonna take the source variables and multiply them by the transport variables. And that's gonna give us our P index score. And this is the state in which most states are at. Most states are in this multiplicative version of the index, source multiplied by transport. Now there's some states that have moved beyond, and this is basically the difference between the old Maryland Phosphorus Site Index and the new Phosphorus Management Tool. So all they did was split apart the source variables and split apart the transport variables and multiplied the appropriate ones together, and then summed them up to get your final P index score. And literally that's all that happened. No variables have changed, just changed the way the numbers are being multiplied together. But it gives us a better idea, again, which one of those transport pathways is our issue? Particulate, runoff, or subsurface? And then helps the producer, the decision maker, make a decision on management and how to better target it within that to decrease your loss of phosphorus. And this is an example. This is some data from Josh McGrath that was unpublished. He did it out at the Y. So this is the same field, moderate soil test P, same source of soil, same source of poultry litter applied. Obviously, it's the same field, so you'd assume that the transport pathways are the same. It got the same amount of precipitation, right? But when we look across the bottom at these different types of tillage, chisel plow, strip till, turbo till, and no-till, and we've got measured phosphorus loss on the y-axis here, we see that there's a weather and other reasons. Management really was more important than source and transport in this field, right? Everything was the same. It was the same field. Tillage was the only thing that was different, okay? So when we talk about our overlap of that source and transport, we need to make sure that we're also including that management as well. So when we visualize, I love visualizing things. When we visualize the PMT versus the Maryland P index, this is what it looks like. We have each one of these little Venn diagrams with the source, transport, and management for that loss pathway. And so what this does numerically, if you're in an area like Western Maryland where we know that erosion is your issue, those other parts don't even matter. And you're taking into account your erosion losses, and you're changing your management to decrease that particulate P loss. Okay? And this structure, this calculation of this component P index really allows our calculation to take into account the differences across the physiographic regions. So when we're looking at conditions like Pennsylvania, we know that erosion and runoff is dominating that P loss, versus when we're looking at the Eastern Shore and we know that subsurface is really where most of that loss is. So we've just changed around the calculation to be able to take this into account. All right, I'm going to really quickly go through some of the results from the drawdown study. Don't worry. Okay, so I gave these talks last week. We were at our scientific meeting out in Phoenix, and it was all like rigid, and they made me stand behind, the podium, and I had to talk into the microphone the whole time. But I tried to make them a little more interesting and sum up the highlights for you guys, because I knew you didn't care about all the other stuff I was going to say. All right, so we had 3 locations across the state. 1 on the Western Shore, 2, we got the Y and Poplar Hill. Those were the soil types, different soil types across the state. Split plot, randomized. We had 4 reps at each site, so, we think we did it right. We had an initial P loading stage. So we took different types of manure at different rates to basically jack up the soil P concentration. All right? And the goal was to create plots that had significantly different soil P. All right? So we looked at the effect of soil P concentration, and then we planted replicates, okay? So we were looking at the effect of 2 things, crop system and initial soil P concentration. So during this P loading phase, we applied all the phosphorus, and then we stopped after that. Now, throughout the duration of the study, we applied other nutrients and lime based on our soil test results, but no more phosphorus got added after that initial P loading phase. These were the years we took this every other year. There were a lot of people that took these samples, so some years they got missed. We did Malic 3 extraction. At harvest, we took all the above ground biomass from the forage plots, but we only took the grain from the grain plots. We did biomass and moisture, and we got the plant tissue P concentration, because we're looking at how much phosphorus is being taken up by those plants, okay? All right, another one of those, don't make your slide with this many words, but the point of this was the rotations differed slightly across the locations, all right? So the grain rotation basically stayed the same, corn, wheat, beans in a 2-year rotation. That was pretty consistent across the 3 locations. But some, like the Y and Simrec, there were a couple years where there was multiple harvests of alfalfa as part of the rotation, but down at Lesrec, down at Poplar Hill, it was basically just silage and rye haylage year after year. There was no alfalfa. So because of that, the rotations were different. So statistically, we kept all the data separate by the locations. We didn't lump them together because we felt that the rotations were different enough that we should really keep them apart. So everything I'm going to show you, you're going to see each of the locations, all right? Okay. We've got our locations here. The X have the different manure treatments, all right? So increasing initial soil phosphorus, all right? And then on the y-axis, amount of plants over. So we just phosphorus removal. And basically what we found was the blue forage plots removed a lot more P than the grain plots. So there was the trend that as we had increasing initial soil, is that what I said? Yep, forage systems remove more P than grain systems. Boom. That's what we saw. All right. Then we looked at the soil concentration over each year, the duration of the study. Those, and we drew these downward lines. We're looking at how quickly, how much phosphorus each year was being pulled out of those plots, all right? So there was an effect of crop the rate at which phosphorus is pulled out of the soil between grain and forage systems. The forage plots had faster drawdown rate than the grain plots. It ranged from 2.9 to 5.4% per year. That's how much is being pulled out per year.
Well, so the other issue with that was he was
But
money from the company to do that.
only at that research center did we see. So up until this point, I'm showing you here, what this is telling you is that that's what this shows. I'm telling you at one location it was important. One location, and we only saw a difference here in the first manure treatment that was not control. Drawdown rate dependent on initial soil P concentration, but only for one soil concentration. Right. Next slide. Published. I wasn't going to make these slides, but the reviewer said I had to, so I did it to make them happy, right? So we plotted how much that soil P decreased from beginning to And when we look at these, I hope you can see, as I can see, this isn't pretty. There's not a very strong relationship here. You can look at the equations for the line, but I'm telling you, these are not significant, okay? There was a pretty weak relationship between crop P removal and change in soil P concentration.
The organic product isn't really going to
Conclusion. We don't think it's free. All right, we should be looking at a form of phosphorus. Giving us the full picture here over as that phosphorus is being drawn out of the soil. Remember, back to the yay, back to phosphorus. Other forms of phosphorus being by 3. We were missing all this other stuff by only measuring what was happening in Malik. Here, the folks at the University of Delaware are looking into, we saved the soils. We had some forethought. Let's save these soils. So we are going back now and looking at some fractionation and seeing if maybe we can find a better relationship, or some of the other P pools can just tell us what's going on. What's happening as we're drawing that P out of the soil? What's happening to those other P pools? Okay? So we took those equations. This is the important stuff. This is what everyone wants to know, right? If I've got high P soils, how long is it going to take before I'm down below the level and Maryland's going to let me apply P again, right? You don't have to admit it, but I know that that's what you're asking. So we calculated based on those lines. What'd you say? What? Okay, it's all right. You can leave me out of it. All right, 18 to 44 years, depending on where you were, to come back down to that optimum. I chose 100 as optimum, so it may be a little less if you're going to go with 150. 18 to 44 years to get back to optimum Malik 3. That's a long time, right? Yeah. So those of you who may not yet be in that situation where you're being restricted from P application, heed the warnings. Don't put yourself in that situation, because soil P drawdown is a long, slow process. Oh look, that's what I said here in my conclusions. P index is necessary for assessing the risk of phosphorus loss, right? Soil test P doesn't work. I hope I've convinced you all of that. Soil test P is not enough. You need the P index because it accounts for site-specific source and transport processes. The PMT is just an updated version of the P index. It accounts for management and different geographic regions. And the long-term drawdown studies suggest soil P drawdown is a long, slow process. It may be expedited if you switch to a forage cropping system. How desperate are you? You want to draw it down a little faster?
I don't ever know.
Up your rotation. But I think that future research, we really need to be working on those different forms of soil P and how they could be playing into this whole decrease in soil P drawdown. Did I run over time? Ah, I did. Thank you all for your attention. I appreciate you staying. I'll be here for a few minutes if you want to ask me questions, but I'm going to let you guys go. Thank you.